Thank you very much, Mariel, for that very nice introduction. And I really appreciate this invitation, especially being here in the fall. It's so gorgeous. And thank you for your time this next hour. Now, I think most of the people in this room are probably aware of this idea that early life environments and experiences beginning even prior to birth have effects across the life cycle on health and human capital. And I think David Barker, epidemiologist from the UK, gets a lot of credit for promoting this idea starting in the late 80s. Now, he and others started finding relationships like this here. You see that coronary heart disease, heart attack rates increase as you go from high to lower birth weight, right? And this is the opposite of what we typically expect, right? You associate heart disease with overnutrition, being overweight, that type of thing, right? So he proposed that undernutrition in utero was leading to those same kinds of conditions. And at this point, this set of findings has been replicated numerous times. So here's a meta-analysis of 135 studies, almost 8 million people, and you can see that same inverse relationship is there, okay, across the full birth weight distribution. Now, the rate does increase at the highest birth weights as well, right? And that's a completely separate pathway that involves the effects of overweight and obesity, the sort of intergenerational effects of that. Very important, but separate from what I'm going to talk about today, which is the left-hand side of this curve, reflecting the effects of kind of gradients of fetal undernutrition long-term. So we now know that prenatal nutrition has effects on just about everything, right? Growth rate, blood pressure, how our body handles nutrients and metabolism, immunity, and so on. And we have very good animal model work to back up a causal role of nutrition in these relationships. So I'm just going to walk you through one example of this, right? Because of course, like birth weight is a very crude measure. We'd like to have some causal evidence that this is actually what's driving the show. So what this shows here, see if this pointer works. It doesn't. Maybe this will work. Yeah. So here, yeah, it doesn't work. On the left, the line there, that's the fasting condition. All right, so that's fasting glucose. Then there's a glucose challenge. So glucose spikes up, and then the bodies start to, generate insulin and remove that glucose, right? So it comes back down. Now, the open circle animals, their mothers had their diet restricted by 30% during pregnancy, right? And you can see that they're clearing their glucose just fine, just like everybody else. But if you look at the insulin, the story is very different. So in the fasting condition, their insulin is much higher. And then after that challenge, their insulin spikes up much higher than everybody else, right? So they're clearing their glucose, as you see on the left, but their bodies are having to work harder at it. They're having to overproduce insulin to clear the same amount of glucose. What this tells us is that their tissues are resistant to the effects of insulin. There is insulin resistant, which is on the path to diabetes. So this is the kind of finding in animal models, which is nutritionally driven, that gives us some confidence that those birth weight relationships, those very kind of crude observational data that we have in humans, actually reflects in part an effect of nutrition. And this makes sense, right? Because development is, you basically start as a single cell, and then you go to 2 cells, and then 4 and 8, and so on. And all the systems in the body and all that kind of regulatory complexity has to emerge from that single cell. And so early in development is naturally going to be more sensitive. There's just more that can be modified, right? And so we know that there are sort of different critical periods in utero and different systems and organs and so forth are most sensitive. And that sensitivity, of course, does not end in utero, continues after birth. There's a lot of growth in the brain in the first couple of years of life, obviously a lot of cognitive development that's happening and so on. And so this literature has really inspired this concept that many of you have probably heard, this idea of the first 1,000 days. This is a way of sort of packaging the findings from this work. And it's the first 1,000 days starting from conception, right, through birth, and then the 2 years after birth. That's the first 1,000 days. And the idea is that this is a critical period that we need to target in our interventions. And if you have never heard of this, just Google it. You're going to see that there's a million organizations that have latched onto this idea as a sort of policy orientation, right? And so here's one organization, and I'm just, there's a real focus here on nutrition in a lot of this literature and these efforts. Nutrition in particular plays a foundational role in a child's development and her country's ability to prosper. Poor nutrition in the first 1,000 days can cause irreversible damage to a child's growing brain, affecting her ability to do well in school and earn a good living, and making it harder for a child and her family to rise out of poverty. It can also set the stage for later obesity, diabetes, and other chronic diseases, which can lead to a lifetime of health problems. So that sort of encapsulates the idea. the Gates Foundation, which has an enormous impact on sort of global priorities, for sort of public health policy, has also sort of organized its nutritional orientation around this, right? So they say we invest in nutrition to reduce preventable deaths and improve maternal and child health, with a particular focus on the 1,000-day window of opportunity from the onset of pregnancy to the child's second birthday. Nutrition is the key to ensuring that individual not only survive, but they can truly thrive, et cetera, right? And then many of you will probably be familiar with the Heckman curve, Jim Heckman, showing this idea that investing early, you're going to have increased returns in terms of human capital in the long run. And notice that he has the prenatal period as the period when those returns might in fact be highest, right? So there's not only this concept of the first 1,000 days being important, but the prenatal period, given the amount of development that's happening and the sensitivity of that development, is recognized as especially important within this period, right? Now, the argument that I'm going to make today is that fetal nutrition does in fact have widespread effects on many outcomes, a lot of the stuff that we just talked about. However, altering the mother's macronutrient intake, and by that I mean protein, carbohydrates, and fats, so the bulk of what we eat, not vitamins and minerals, not trace nutrients. Altering the mother's macronutrient intake during pregnancy has minimal effects on fetal nutrition, growth, or offspring health in humans. There are good evolutionary biological reasons for a lack of strong intergenerational effect of pregnancy nutrition. Fetal nutrition may be more related to the mother's nutrition prior to pregnancy than to nutrition during pregnancy. And then the punchline, I'm going to sort of steal some of my own thunder, is that harnessing DOHAD to improve health will require improving nutrition well before the 1,000-day window. Targeting nutrition during pregnancy itself is not a shortcut, or it's not a magic bullet to meaningful improvements in health and human capital. Right, so that's my argument, and I'm going to make that argument by walking through a couple of different literatures. First, we're going to look at evidence for buffering of the fetus from the mother's experiences nutritionally, and we're going to first focus on this famous example of the Dutch famine, and which is a kind of gold standard study that many interpret as evidence for an intergenerational effect of nutrition. And then we're going to look at the other side, nutritional supplementation. Is there an effect there on the next generation? Then we'll talk about sort of interpreting what I'm going to show you, which is a lack of effect in both of those, really, or very weak effects. And in this here, we're going to focus on the evolution of buffering strategies and how they've taken a very kind of elaborate form in humans. And then we'll finally think a little bit about the limitations of that buffering. So when we think about nutrition, there's a lot of buffering, but in fact, the mechanisms that ensure that delivery of nutrients can have collateral effects, and especially cortisol. So there are intergenerational effects there, and we'll have a few implications at the end. Okay, so to get started, how strong is the evidence that maternal nutrition during pregnancy influences offspring health outcomes in humans? And I'm going to argue not as consistent as we often like to think, or as conveyed in this literature. Now, first of all, we've already seen this, right? That most of the evidence from this DOHAD literature as applies to humans is observational. It's using these kind of indirect markers like birth weight, right? Which do tell us something about fetal nutrition, but they also tell us about genetics and whether the child is born into a low SES household, all sorts of factors that could independently influence health in the long run, right? So it's very confounded. Quasi-experiments are, of course, more causally informative. And the Dutch famine or hunger winter studies are the most often discussed and cited as the gold standard in this field. And I'm going to walk through this in a second. A benefit of this paradigm is that multiple groups have conducted these studies in multiple cohorts. So there's some replication. We can look at how consistent or not the findings are across those studies. Now, if you're not familiar with these studies, what they are is they're follow-ups of individuals who were exposed to famine in utero during World War II, when the Nazis blockaded this region of the Netherlands. You can see this graph on the right is showing rations, and you can see they go way down. The caloric intake per day goes way down during this period. And again, there have been multiple follow-ups. The big ones are based out of Amsterdam in the Netherlands, and the other is based out of Columbia in New York. And those are the ones I'm mostly going to focus on. Okay, we'll have a little dabbling in a few other studies as well. Now, I imagine just about everybody here has heard of the Dutch famine studies, right? They're extremely famous. You have these headlines like "Mom's environment during pregnancy can affect her grandchildren" in the New York, and in Science. Carl Zimmer in the New York Times a few years back. The famine ended 7 years ago, but Dutch genes still bear scars, right? That kind of coverage, there's been a lot of it, right? But how strong and consistent is the evidence that prenatal exposure to the Dutch hunger winter actually did negatively impact later health? Now, I've been interested in this for a long time because I started really engaging with this literature in the mid-90s when I was in graduate school. I wrote my first grant in 1997 to basically test what at the time was called the Barker hypothesis, right? That the earlier permutation of DOHAD in the Philippines. And I'll talk a little bit about that study in a bit. But I was writing up my my findings in my dissertation in 2000, and I was focusing on cholesterol metabolism as an outcome. And I was really excited. This study came out from the Dutch famine in 2000. And if you read the conclusion, an atherogenic lipid profile might be linked to a transition from poor maternal nutrition in early gestation to adequate nutrition later on. This suggests that maternal malnutrition during early gestation may program lipid metabolism, et cetera. Exactly the kind of study that I needed to study to cite in my dissertation and make my case, right? And here's the findings. So you can see that there are many different tests here. Okay, different periods of gestation, and basically one of these coefficients reached significance, and it's the ratio of LDL to HDL. So it's just this ratio measure was elevated in those who were exposed early in gestation. And so I cited that. Here's this evidence that there's a long-term effect, but obviously it's pretty weak already. You can see that. This is the Amsterdam-based study. Now let's look at the Columbia-based study, and you can see here that females, there actually was some evidence for elevated risk if you were exposed to famine in utero and with a reasonable sort of effect size. But if you look at the men, there's kind of comparable sized benefits, right? So quite murky. It's either beneficial or negative, depending upon whether you're male or female. So not a consistent effect. How about blood pressure? This is the Amsterdam-based study. There's nothing highlighted here because there were no significant relationships. Okay, so that's the Amsterdam-based study. Now let's look at the Columbia-based study. The top row is unadjusted, and you can see that the confidence intervals just barely don't encapsulate one on the right, et cetera. So these are just very marginal findings, and then when they adjust, they're no longer significant, right? So not a whole lot here either. Right. This is a review written by one of the PIs of the Columbia-based study, and they kind of outline the literature as a whole. And they say of all reported outcomes, associations between prenatal famine and adult body size, diabetes, and schizophrenia show the most consistent pattern. For other outcomes, the pattern is more variable and inconsistent. So let's focus in on diabetes. That's one that it's arguing is relatively consistent. So here's the Amsterdam-based study. Fasting glucose, the most basic, the clinical measure of diabetes that every doctor in America uses. P-value of 0.99. Okay, not great, not basically nothing there. Insulin, HbA1c or glycosylated hemoglobin, also not there. After a 2-hour challenge, there was some elevation among those who were exposed to the famine, sort of marginal. But then if you look at rates of diabetes themselves or impaired glucose tolerance, very non-significant findings, right? And then here, how about coronary heart disease? This is sort of the endpoint, why all this stuff matters, right? This is, diabetes, hypertension, all these things lead to heart attacks, right? And there is one significant point here, early gestational experiences, slightly elevated incidence, but you can see that the cell size is 11 individuals. So just moving a few individuals is really going to change those findings. They're not robust, right? So I think the findings here are actually not, they're not as we typically hear them conveyed. They're actually quite patchy. Now, what about effects on cognition, right? This is super important for understanding capital formation. And here's a study that was published in the Proceedings National Academy in 2010. Here are all the various tests they did. And of those, there's one that reached significance. This particular study, they did not Bonferroni correct for multiple comparisons, so I'm not sure that this even passes muster, but it's a single measure of Stroop test that shows some evidence of being declined in early gestational exposure. Here's another study. This is the Columbia-based study. I'm almost done with all these studies here. So that was the Amsterdam study. Here's the Columbia-based study. Individuals exposed to famine during gestation do not differ in cognitive functioning at mean age of 58 years compared with individuals born before the famine or conceived after it, or compared with unexposed same-sex siblings. All right. Now, let's go back. There were some very early Dutch famine studies that sort of preceded DOHAD and Barker and all that. This was from the 70s. They looked at 125,000 19-year-old male recruits, so a very homogeneous population. And they looked for evidence for cognitive effects of being exposed prenatally. The authors conclude the findings point either to a high order of protection afforded the fetus in utero or to great resilience of the fetus in the face of nutritional insult or both, because they found nothing. All right, now when I was researching all of this and sort of trying to compile what we knew about these studies, I came across this fascinating interview with the author, Notestein, of this article here. He was sort of a retrospective on her career. And the interviewer asks, our data don't always support what we might believe is the best for public health. Has this ever happened to you? Notestein, yes, with the Dutch famine study especially, we found no evidence that 6 months of starvation and pregnancy among a reasonably well-nourished population had any effect on the intelligence of offspring. Many of our colleagues were furious with us because it would have been much more satisfactory in terms of social justice to have found that food did matter, but in fact, nothing has happened since to change that opinion. What we have learned is that certain nutrients, particularly folates, can make a difference depending on timing and outcome, but not general starvation in any direct way. So I think with the Dutch famine, we have a bit of a paradox. On the one hand, we know from the Doehead literature that lower birth weight is inversely related to all these outcomes, heart disease, hypertension, et cetera. And we know that animal models, you restrict the diet of pregnant mothers and you replicate those findings, right? So we have mechanistic, good experimental evidence to support the idea that fetal nutrition is actually driving those relationships. And yet offspring of mothers, human mothers that are exposed to fairly severe famine while pregnant, often show very inconsistent and arguably, in some cases, no relationship with those same outcomes, right? This is very different than the way that this literature is typically portrayed. And I think that, it's underappreciated just how patchy these findings really are. So that's the example for nutritional stress. I think there's a lot of evidence that that's not getting through to the fetus. What about the other side? If we supplement diet, improve women's diet during pregnancy, there have been efforts to do this going way back, way before Doehead, way before Barker, to try to improve birth outcomes. And here is a systematic review, Cochrane database, and they kind of grouped the populations based upon the adequacy of the nutrition of the women that were in the study. And the 5 adequately nourished women studies, you can see there's a roughly 1 ounce increase in birth weight of these interventions, but the p-value is really not significant or really close. And then they looked at 9 studies that involved women that were undernourished, and you see a larger effect size and approaching a significant p-value. Now, if you dig closer into this and look more closely at it, there's actually one study in particular that's driving that increase in effect size in the undernourished group. And if you pull that out, the effect size becomes almost comparable to the adequate nourished woman, and it's very non-significant. That study is a study it's a very famous one in the Gambia. It's called Canaba. And if any of you have any sort of interface with the world of maternal child nutrition and health, you'll have heard of this study. It's been around for, I think, 70 years. It's an MRC study, so it's funded by the UK government. It's been there forever. And what they managed to do is they achieved a net 431 calorie per day increase in intake for 4 years in women in this village. Then they compared birth weights during the 4-year intervention period to the preceding 4 years. And they were successful in raising birth weights during the hungry season, during the wet season, but not during the rest of the year. Let's take a closer look at these findings. So in the wet season, you see that there's an increase in birth weight, pretty substantial, but in the dry season, there's nothing, right? Now, what's going on in the wet season? In this population, it's a period when food supplies are low, where women are having to work in the field for the harvest. And when you have it, because it's wet, there's more infectious disease. So it's sort of a triple whammy energetically. And so it's a big nutritional drain during this period. So much so that this is showing the rate of weight gain. You can see that early in pregnancy, women who are pregnant are actually losing weight instead of gaining it. Okay, so this is severe nutritional stress. And so what I think the Gambian findings tell us is that if you have a substantial increase in nutritional intake at a period when nutritional stress is so severe that women are actually losing weight rather than gaining it while pregnant, right, you can actually have a fairly substantial beneficial effect on birth outcomes. But that's a fairly severe situation, right? And it's really not all that surprising. During the rest of the year, and in all of those other studies, in that database, there was effectively no effect of those supplements on birth weight, right? What about long-term health outcomes? Of course, the folks at Canaba are aware of this literature, and so they've looked at other kinds of outcomes as well. And you can see here that they found that being exposed to that supplement in utero may have little effect on cardiovascular risk in their offspring. And we've shown little evidence on cognitive development, right? So they actually did not find any long-term effects of that supplementation. What they did find is that there were substantial effects on the women themselves, right? So there was an increase in their fat deposition. They had improvements in their energy level. There were comments about how when they were working in the field, they were like more likely to be singing and in a like good mood, right? From the extra energy. And their bodies did allocate more energy to reproduction, but it wasn't to the current baby. You don't get this increase in the size of the current baby. What you got was a shortening of interbirth interval. So this is a naturally contracepting population. And when lactating, that suppresses ovulation, right? And if you're energetically stressed, it'll suppress it for longer. So if you improve nutritional status, that lactational amenorrhea will be shorter and you'll get a reduction in interbirth interval and fertility will go up. And so this was an increase in fertility of about 37% as a result of that supplementation, right? So not increasing birth size, but increasing the number of kids. That's what came out of this. And if you're interested in this topic, Meredith Righteous's piece in Science from 2019 is a really brilliant analysis of this work that I would highly recommend. Okay, so the Dutch famine and the macronutrient supplementation work, I think, show that large changes in what the mother's eating in terms of her protein energy intake during pregnancy, whether it's a famine on the one hand, shortfall, or supplementation on the other, often have minimal effects on the fetus, their growth, or their long-term outcomes, right? And this is not how we think about this in the Doe-head literature, but I think it is what we should expect. We should not be surprised by this. And I've already alluded to some of this. This process of early development is exceedingly complex and very, very fragile, right? There's a lot going on early in life, and the solution to that evolutionarily has been to build up elaborate buffering strategies to make sure that it's cushioned. Right? And if you look at the history of life, vertebrates from fish to us, you're going to see that there's a progressive movement towards increasingly buffered and increasingly buffered early development. So in fish, of course, they just kind of lay their eggs out in the water, spawn, no parental care of any sort. It's just whichever eggs manage to survive. Amphibians and reptiles, you start to get some internal fertilization. But really, it's more akin to the fish model with minimal investment. With birds, you start to get real investment, right? Because now you're laying eggs in a nest, you're incubating and maintaining a constant temperature. When the chicks hatch, there's protection and feeding of the chicks, right? So there's a lot of kind of protection and buffering of that rearing environment that happens in birds. Marsupials, the earliest mammals. Right? So now you have internal fertilization and live birth, and then you have early development that's being sustained off of breast milk, off of the mammary gland, internally within that pouch, right? And then you get to us, right? Placental mammals, where now the baby is attached to the mother's circulation directly through an umbilical cord and placenta on top of all those other things, right? And so as a result of this, the first 9 months, right, from conception until birth, you have constant temperature, stable nutrients and oxygen delivery, pretty good protection from pathogens. And in our case, as I'll talk about in a second, our placenta is invasive. And this gives the fetus some control over the mother's metabolism and actually helps ensure its delivery of nutrients. And I'll get more to that in a second. Okay, but I really want to emphasize this word metabolism here because metabolism is key to this buffering. So let's take a kind of a deep dive into what's going on metabolically that helps buffer the fetus. Let's forget about the fetus for a second. Every cell in our body constantly needs energy to stay alive, right? Like I'm sitting here talking to you, I'm not eating. I haven't actually eaten in a couple hours. My body is moving resources around within my body, right? To keep everything going. There's a constant supply of glucose in my bloodstream and so on. And that is facilitated by the fact that I have nutrient stores. I have body fat, I have glycogen. And then there's an interconvertibility. So different nutrient classes could be converted into one another to redress any sort of imbalances, right? So I'm going to focus on glucose because I'll explain why. It's very important for fetal development. But this right here, this dizzying chart is human metabolism, right? And it is extremely complicated. If we focus in on a little piece of it here, right? We zoom in and up on the upper left here, you'll see that there's glucose. And down here, you see that there's fatty acids. And if you have a lot of time on your hands, you can actually trace through how When you drink a Coke or a Sprite or something, your body takes that extra energy and converts it into fat and deposits it in your adipose tissue, right? This is how it's doing that. And it works in reverse, right? When you're not eating, you can mobilize that fat and convert it into glucose and use it in the body, right? So metabolism is extremely flexible and capable of moving things around. Why am I focusing on glucose? Well, these are very crude data. These were data that were not even collected for this purpose. This is from our Philippine study showing that a mother's fasting glucose levels while she's pregnant is a predictor of birth outcomes. And this is very well known because it's the gradient between the mother's glucose and the fetal glucose that determines how much glucose passes across the placenta. And the glucose in turn is stimulating insulin, and growth in utero is insulin driven, right? So it's like a very direct connection between the amount of glucose that the fetus receives and its growth rate. That changes later, after birth, but in utero, glucose is key. So let's take a closer look at what actually is regulating glucose levels, given how important it is. Of course, in some sense, it's a reflection of the mother's dietary intake, but after you wait a couple hours and then you start kicking into your glycogen stores. You mobilize stored sugars. And then when those are depleted after hours, you can break into these longer term stores like body fat, right? So body fat is broken down and some of it becomes glycerol. Glycerol goes into gluconeogenesis. This is the production of new glucose in the liver. And some of it enters the bloodstream as free fatty acids. When your free fatty acid levels go up, your muscle will preferentially take that substrate up and use it as energy and become insulin resistant. What this does is it spares that glucose, which is prized for things like the brain and during pregnancy for the fetus. All right, so there's a glucose sparing effect of that. And then if you really are starving for a long period, you can even metabolize, your protein, like your muscle and organs and so forth. And that can also be a source of glucose, right? So glucose is super important for the fetus. The level of glucose that's reaching it is actually what's driving growth. But you should see here that feeding the mother more is not necessarily going to change the glucose levels, because what's regulating glucose is this entire system. It's a homeostatic system that's maintaining stability. And food is just sort of entering in as a currency that goes into storage, can be mobilized and so forth, right? So placental mammals have evolved very elaborate systems to buffer early development against the vagaries of maternal experience during pregnancy. They've done this by bringing early development within the umbrella of this metabolic, very internally regulated, stable system. But even compared to other mammals, humans appear to be particularly effective at this buffering, and there's a couple reasons. One, which I alluded to, is we have an invasive, what's called a hemocorial placenta. And typically you see this only in small species like rodents, but we have it. And what this allows is the human fetus to secrete hormones directly into the mother's bloodstream. And there's no way for the mother to stop this, right? So those hormones in turn can have effects on our metabolism. So placental lactogen is secreted by the placenta. It makes the mother's tissues insulin resistant, which, as I just talked about a second ago, that reduces uptake of that glucose by your muscle, and it spares it for the fetus. So when the blood goes through the placenta, now it has higher glucose levels, and more of it passes across the placenta to the fetus, and growth is increased, right? So this gives the fetus some level of control. And then other factors that I think increase our buffering compared to, say, the mice and rodents and stuff that are studied in the Stohead literature. We have a larger body size, which means greater buffering for many reasons. We're more efficient energetically and so on. But we also have a much smaller ratio of offspring side to adult body size, right? So if you look at mice here on the left, the size of the litter is about half the weight of the mother, right? I mean, you can imagine how horrifying that would be if human women gave birth to a 60-pound kid or a 50-pound kid, right? That's what's happening in mice. And so you can imagine that there's very minimal capacity to buffer pregnancy in a species like that compared to us, where it's just a small fraction of the mother's metabolism and body weight. And Zane Thayer, former student of mine, we compiled data from the literature. So the pink lines here, or the pink bars, are the effect size of the Dutch famine on these various outcomes. And then we tried to find animal experiments that had a comparable level of nutritional restriction. And you can see very clearly that there's a body size gradient, right? The smaller species have a bigger effect of the same magnitude of nutritional stress in utero. And we have the smallest being the largest, okay? So I think all of this gives us some insights into why nutritional interventions fail to impact birth weight. And I think it also gives us insight into why the Dutch famine data are as weak as they are, right? All tissues in the body require a constant supply of nutrients. These are supplied through the diet, but also backed up by a flexible and responsive metabolism that manages large onboard energy reserves. Circulating nutrient levels are relatively stable as a result of this. And then the fetal placental unit is embedded within this system, this homeostatic, internally regulated environment. And this buffering is especially effective in humans owing to these characteristics that I just talked about, the mode of our placentation and the relatively small size of our offspring relative to the mother. So I think these buffers show why changes in the mother's macronutrient intake during pregnancy have modest effects on the offspring, right? Now, is this to say that nutrition doesn't matter for fetal growth? No, obviously it has to be getting through somehow, right? And so what is it? If it's not the mother's intake during pregnancy itself, how does nutrition have effects on the fetus? And first of all, micronutrients can be very important. So these are things that have to come from outside the body, like iron, for instance, that has to come from the environment. It's required in trace quantities. We have stores, and those stores are finite and they can be depleted. And so in some contexts, micronutrient supplementation can have a fairly substantial effect. I think there's growing evidence that the periconceptional period, right around conception, could be important because there's a lot of epigenetic sensitivity. So there's some interesting stuff that's happening there that might make that kind of uniquely sensitive. And there are some studies that would point to that. But I think this is relatively understood at this point. What we have known for a long time, however, long before the Doe head literature, is that a woman's body weight, her BMI, going into pregnancy is an important factor, right? So women with higher BMIs tend to give birth to larger babies. And this is reflecting her nutrition chronically for years prior to pregnancy, right? And especially if you think about a place like where I work, where the Philippines, where people are sort of more marginally nourished, someone's BMI coming into pregnancy really is a reflection of chronic nutritional availability, right? And so that is an influence. And then increasingly, we have more and more evidence, I think, that the mother's own early developmental nutrition has an influence across generations, her infancy and childhood. So I'm going to focus on that. And I'm going to show you, we're going to come at this from a couple different angles. So the first one is we're going to look at the mother's leg length as a proxy for early nutrition, and I'll explain this. Right, the hypothesis is that the mother's leg length will be a stronger predictor of offspring birth weight than will her trunk length. The rationale is that leg growth is fast in response to nutrition during infancy and early childhood, whereas trunk growth is slow and less nutritionally sensitive. It's considered to be a little bit more genetically catalyzed as a trait. And this method is used a lot in anthropology. It's sort of a retrospective marker of early nutrition. Let me give you an example of where this idea came from. So in Japan, post-World War II, there were big changes in diet, and there were increases in stature, entirely driven by changes in leg length. All right. So Japanese population was getting taller because they were growing larger legs. So the idea is that as nutrition improves, population gets taller, trunk basically stays the same, and legs get longer. All right. So it's that ratio that can give us some insight into what was going on early in life. Now, this study was actually, so I work in Cebu in the South. I'll talk about it in a second. This was actually an undergraduate project done outside of Manila in a place called Antipolo City. This is the undergraduate. He was a psych major at Northwestern about 10 years ago, Green Chung. He had a connection to this birthing clinic, and we designed the study together. He went there and implemented it and did a wonderful job. And these are the findings. So basically, we're looking at birth weight. He did anthropometry in the mothers, and we just hypothesized that leg length would be a stronger predictor. And of course, you see that it is, right? It's about 15% of the variance explained by leg length, very little explained by trunk length. And when you look at the gradient here across tertiles of leg length, there's a pretty substantial association, right? And this is adjusting for trunk length and other factors as well, right? Now, this is very crude, obviously, and we'll get to other data here in a second. What I want to do now is shift from Antipolo down to where I work, which is in Cebu. And I've been working there for 26 years, I realize now. This is a study that began about 40 years ago in 1983. It's been tracking the same, enrolled more than 3,000 women who were pregnant at the time, and now been tracking their offspring and their grand offspring. So I've been working with the study since, I guess, 1998, when I went there and lived for about a year and a half, and been working there since. So we're going to use some of these data on generation 3 birth weight and relate that back to what was going on with the mother and a teeny bit to the grandmother as well, right? And if you are looking for causally informative data, close your eyes because we don't have any. Okay, this is all observational, correlational. So bear with me with that, but this is just showing what I was talking about a second ago, that a woman's BMI going into pregnancy is a predictor of birth weight of the baby. This is adjusting for the mother's own birth weight, right? So this is independent of that. It's adjusting for her stature, right? So this is something that's been replicated many times. So it suggests that nutrition in the years prior to pregnancy is an influence on the nutrients that the fetus receives and the size that they're born. Right, but what about her own early life nutrition? I flagged that as something that we want to focus on here. What this shows are patterns, and of course, this study began right when the offspring were in utero when this study began. So we have data going all the way back into the earliest environments. This shows exclusive breastfeeding rates. So they start off high and they drop off by six months of age. And in parallel, you get an increase in diarrheal illness. And this is something that you see in populations like this all over the world, right? As weaning is happening, illnesses increase, especially diarrheal illness. Right? So those are related. And in parallel, you get an increase in the prevalence of stunting, right? So nutritional stunting in a population like the Philippines in the 1980s was primarily driven by weaning stress and infectious disease. And this is something we know, right? And so here, this is interesting because stunting by 2 years of age is sort of a cumulative reflection of that nutritional stress in the years prior, right, in those first 2 years of life. Here's an analysis that pooled our data from Cebu, along with Pelotas in Brazil, Delhi in India, and the INCAP study in Guatemala. And you can see that maternal stunting at 2 years of age is a significant predictor of low birth weight, and inverse predicts a reduction in birth weight as a continuous measure as well, both significant, whereas the father's stunting does not. Right? So that would argue that this is probably not a genetic story about genetic shortness, but something about specific to the mother's nutrition driving that stunting that in turn has an intergenerational effect on the next generation. Right. So this is all observational. One more experimental data point, pretty famous study from INCAP in Guatemala. This is back in the 70s. They randomized 4 villages to different diets. So one was a high quality diet that had both protein and energy, and the other was a lower quality diet, just carbs. And then they fed these for 7 years, and then they looked at various outcomes. And what they found was that girls that were given the higher quality diet as children gave birth to substantially larger babies compared to the folks from the villages that got the lower quality supplement. And they also saw effects on things like birth length and head circumference. So this is getting closer to experimental evidence. All right, to summarize here, nutrition, I think prior to pregnancy and during the mother's own early development seemed to be stronger and more consistent as predictors of offspring birth weight in humans. That, the diet during pregnancy itself is not super important, at least with respect to macronutrients. Now, I think all of this suggests that efforts to harness DOHAD biology must focus on nutrition well before this first 1,000 days, right? That there's a lot going on prior and that we need to take that seriously. All right, so everything I've been talking about so far is about how nutrient delivery to the fetus is highly buffered, right? So it's very hard to reach it through interventions and so forth. But there are factors that do get through. And somewhat interestingly, that homeostatic machinery that maintains stability in nutrients is part of what can have negative effects on the fetus, and especially cortisol levels. So let's take a look at this. This is where we'll sort of the last topic, talk about the limits of buffering and the costs of that homeostasis, specifically cortisol. So schematically, you can imagine the mother's experiencing the environment, right? Good season, bad season, good month, bad month, sort of bouncing around. But we already know that the internal to her body, glucose is still being delivered, right? Her heart is still beating, her brain is functioning, glucose is being delivered at a constant rate. The fetus is experiencing that internal constant environment. But what's maintaining that is homeostatic biology, right? There's all kinds of machinery that's moving around, and cortisol is an important part of that. Right. So cortisol spikes during acute food shortage, and the effects of this are to mobilize stored body fat for energy. It reduces skeletal muscle glucose use, which again spares it for other functions, so it's glucose sparing. And it also increases glucose production in the liver. So cortisol has a lot of effects during fasting and starvation. And one example of this, there aren't too many studies, as you can imagine, ethically, of women who are starving and looking at their hormones. But during Ramadan, they've shown that cortisol is elevated among women who are fasting. All right, so why does this matter? Well, cortisol, so you have two different kinds of hormones. You have protein hormones, which are big and require specific receptors to enter into cells. But then you have steroid hormones, which are basically very small lipid-based molecules. Cells also have a lipid bilayer, right? So those lipids pass right through the cell membrane. There's actually no way to stop it. So steroids just passively can go into any tissue. And the way, except the placenta expresses an enzyme, as I'll talk about, that actually converts that active hormone into an inactive hormone. So that's how you get the shielding. And in animal models, fetal cortisol exposure leads to similar long-term health outcomes as fetal undernutrition, right? So high blood pressure, insulin resistance, all these things, effects on cognition, et cetera, right? So very similar. And so on the left is the study I showed you at the beginning with guinea pigs, where they're overproducing insulin to remove their glucose. On the right is sheep. Same thing, except here they're exposed to cortisol, right? The time axes are not the same, but basically they're showing the same thing. One is generated by undernutrition, the other is generated by maternal cortisol levels. So under normal conditions, you can see that blue dotted line. That is the enzyme that is shielding the fetus. But when there's a spike in maternal cortisol or when that enzyme layer is depleted, cortisol gets through and has all of those programming effects on the next generation. And there are a lot of studies in humans that have done the kind of shock design, but where the shock is very clearly not nutritional in origin, right? Where it's clearly psychosocial and that can have lasting effects in utero on things like birth outcomes. So here's exposure to an earthquake in Chile. You can see there's a dip in birth weight that is being driven by prematurity on the right? So there's evidence there. Exposure to a big ice storm in Canada, reduction in birth weight. And they've also done some follow-up studies in this to look at different phenotypes in the kids as they've grown up. One thing that they found, the cohort's still quite young, but they looked at fingerprint asymmetry. And fingerprints are laid down very early in development. So asymmetry is considered a marker of early developmental stress. So this suggests that stress is actually getting through and impacting early development. They found some cognitive and behavioral differences at 5 years of age. And I looked for updates on this study while preparing this talk. And here's a new study where they found differences in things like functional connectivity in the brain and so on. So they're seeing long-term effects of being exposed to that stress in utero. Some of you may be familiar with this study, really kind of an amazing design. They looked at birth outcomes in all counties in California. And you can see that the year after the 9/11 attacks, the risk of giving birth to a low birth weight baby more than doubled, but only among women who have ethnically distinctive Arabic sounding names, right? So this is, I think, interesting quasi-experimental evidence for a role of discrimination in birth outcomes in California. And then finally, the last study I want to talk about, this is my former student, Zane Thayer, who's now associate professor at Dartmouth, but she designed her dissertation in Auckland. She recruited a multi-ethnic sample of women, and she found that as women who reported more discrimination while pregnant had higher levels of cortisol during pregnancy. And then she followed up the kids, so the babies, and got a saliva sample before and after they had vaccines administered at their 6-week pediatrician visit. And you can see the ones whose mothers reported discrimination have much more accentuated HPA sort of cortisol response, suggesting that they're having an accentuated stress response as a result of that prenatal exposure. Okay, so to summarize this, I'm almost done here. The stability of circulating nutrients is maintained by homeostatic biology. That was the first part of the talk. But I think we see also that homeostatic biology itself, which is very dynamic, it has to be, right? It's responding to what the mother is experiencing and doing all kinds of things to maintain that stability and things like nutrients, that biology itself actually has spillover effects and can have effects that do persist into later life. Okay, so what are the lessons here? Let's just wrap this up. Lessons for Doe-inspired nutrition policy. First, I think evolutionary principles underscore the effectiveness of maternal homeostasis and buffering fetal nutrition. This minimizes the impacts of changes in the mother's nutrition during pregnancy, whether it's the Dutch famine or it's nutritional supplementation. Okay, it's going to tend to buffer and maintain stability despite those changes. Okay, and what I think this means for this concept of the first 1,000 days, right? So the earliest period of conception clearly there's a lot of development happening. There's a lot of sensitivity, and there is nutrition is a big driver. But that first third of this overall 1,000-day window, which is most sensitive, is also the most buffered and thus most challenging to influence through nutritional policy. This is a very hard period of development to reach through nutrition intervention. The nutrition that the fetus does receive from the mother is sensitive to her nutritional experiences, but I would argue primarily to her nutritional history. It's her nutrition going into pregnancy and even her early development as well. So the implications of that are that nutritional supplements targeting pregnancy itself are unlikely to have major benefits for offspring growth or health in marginally nourished populations. They will help the mother, and that we need to actually focus on that. Mothers also, their health is important, and so we should prioritize that regardless. But I think that what the Doe-head literature really shows us is that sustained, if you have a population that has had sustained multi-generational undernutrition, there's not a magic bullet. You cannot just target pregnancy itself and reverse that. You actually have to have sustained multi-generational improvements in nutrition. There's going to be a developmental component to that intergenerationally, right? But you can't just target pregnancy itself. On the other hand, I do think that the Doe-head literature and biology suggests that stress alleviation during pregnancy should be a target, that can have immediate effects. So when we talk about adversity, if it's a psychosocial stressor, I think there's real potential benefits there as a policy lever. The very final point, this is my last slide, is I think the Dutch famine gives us a little bit of purchase here to think about these nutritional shocks as a study design and what they tell us. I think that shocks that approximate randomized exposures are, of course, causally informative, but are often mechanistically ambiguous, and therefore ambiguous with respect to policy levers. A case in point is that what I've just walked through, where the modest intergenerational effects of Dutch famine exposure due to insufficient nutrients to sustain fetal growth, the common assumption in this literature, or to the stress physiology that was activated by that nutritional stress, or for that matter, by the fact that they're experiencing war, right? Very different. Negative nutritional shocks always activate stress physiology, cortisol, as a metabolic necessity. That hormonal effect is probably what's having a lot of the effects on the next generation. What that means is that, go to the next point. The effects of nutritional shocks inform us about the cost of experiencing shocks, right, which is not a policy target, but may not be informative about the benefits of improving nutrition in non-shock contexts, right? Because shocks are creating stress, which has the effects, cortisol passing the placenta, inducing all those changes in the fetus. If you have moderately nourished women and you improve their nutrition, it's having no effects on their HPA and cortisol. That entire axis is not a part of that story. And so I don't think it's going to have the benefits that are expected. And that is what I think the data actually bear out. So thank you.